March 23, paper 6. Let's go. Potato cells take up methylene blue dye when the potato tissue is placed in the dye solution. Okay, بيقول لي الفاكت الأول إنه when you put the potatoes in the methylene blue dye, the dye will go into the potato. If the dyed potato cells are placed in water, the dye will diffuse into the water. لو أنا بعد كده I put the potato which was dyed blue in water, الblue dye دي will diffuse out of the potato into the water. A student investigated the effect of temperature on the diffusion of the methylene blue dye from the dyed potato cells. I'm going to investigate the effect of temperature. Then I will change the temperature. This is the factor changed, and then I will see what happens to the diffusion of the dye into the water. The student used this method. You are provided with a beaker of cold water, warm water, and hot water. These are the 8 different temperatures. You don't have numbers, you just have cold, warm, and hot. Measure the temperature of the water in the beaker of hot water. Label one test tube C, W, and H. C, W, and H. Draw a line 9 cm from the base of each test tube. Place the 3 test tubes in the test tube rack. Draw a line أهو, and then put them in the rack. Fill the test tube with the cold water up to the line. Put test tube C in the beaker of cold water. Fill test tube W up to the line and put it in the beaker of warm water. Fill H up to the line and put it in hot water. طيب, what is the error here? There is an error in this way. إيه الerror هنا؟ يبقى here we have 2 errors. The first is we cannot measure the height of water. We should not measure the height of water using a ruler. لا, in water we should measure the volume. You should place equal volumes of water in the 3 test tubes. ده number 1. Number 2, you put the water in a water bath that is not maintained. El water bath ده, if you leave it at room temperature, it will become colder. Our temperature will change according to the room temperature. So you have to use thermostatically controlled water bath. يبقى these are the 2 errors here. You are provided with 3 potato cylinders. They have been soaked in methylene blue solution and then rinsed. Place the 3 potato cylinders on a white tile and cut the 3 potato cylinders approximately 2 centimeters in length. What's the problem here? Actually, أنا هنا I have 2 errors. Yes يا بلال. يبقى as we said, here there is 2 errors. The first error is the rinsing. The rinsing is not standardized. You might rinse one potato more than the other, so one of them will have less dye than the other. يبقى this is a source of error. The other error is that he said approximately 2 centimeters in length. لا, you cannot cut the potatoes approximately. It has to be an accurate measurement, so both of them are exactly the same. Okay. Place one potato from step 8 into each test tube. Start the stop clock. Leave the test tubes for 15 minutes. كلهم سبتهم for 15 minutes, so this is a controlled variable. After 15 minutes, measure the temperature of the water in the hot water again. أهو بيقول لي إيه بقى؟ In step number 2, when you first put the hot water, the temperature was 61. هنا عندي 65 and this is 60. So each 2 dashes are 1 degree Celsius. 61 degrees. طب what about the temperature of the water at the end of the experiment? It was 38 degrees. ده معناه إن it cooled down. طيب Remove the test tubes from the beakers, shake all the test tubes for 10 seconds, place the test tubes into a rack, observe the intensity of the color. What's the source of error هنا؟ Anything that has shades of color, we say that it is subjective. Yes, and the improvement will be use a colorimeter. It is subjective. يعني إيه subjective؟ يعني difficult to judge the color by your eyes because shades might be close to each other. So how do we improve this method? We use a colorimeter. Okay. في الآخر بقى it shows the students notebook with their observation. These are the results at the end. Prepare a table, very easy table. A changed variable on the left and the results on the right. A changed variable is the temperature of the beaker, cold, warm, hot. Results was the color intensity. This is what I was observing. الcold كان pale, هنا medium, هنا dark. طب هي ليه dark? Because the dye diffused out of the potato a lot into the water, so the water became dark. يبقى what is the conclusion? The higher the temperature, the more the diffusion. Higher the temperature, the more the diffusion. Identify one possible source of error in step 8. We already said the sources of error in step 8 هنا. يا إما rinsing is not standardized, you did not rinse the same the three potatoes like each other, or the size of the potato might not be the same, the three potato cylinders, so this is also a source of error. It will have different surface area. لو هما different sizes they will have different surface area. So كده هيبقى عندي two variables changed in the experiment, the temperature and the surface area, and this is not correct. You should only have one changed variable. Identify one safety hazard. What is dangerous in this experiment? That you probably used a sharp knife to cut the potatoes. طب if he asked you what is the safety precaution for this? What do I do in order to protect myself? لا مش wear gloves. The gloves will be cut. Yes, use a cutting board. Use a cutting ثانية واحدة. Use a cutting board. يعني do not cut the potatoes while in your hand. You should cut them on a stable surface. The readings on the thermometer showed that the maintenance of the temperature during the investigation was a source of error. برضو احنا قلنا ان water bath were not maintained. The temperature cooled down. What is an improvement to reduce this source of error? Use thermostatically controlled water bath because it uses electricity to keep the temperature as it is. The student did not repeat the investigation, only collected one set of results. Explain why it is better to collect several set of results. ايه two reasons بتاعت repeat? Why do I repeat the experiment? Increase reliability and identify anomalous results. Okay. A student investigated the surface area on diffusion. The student used this method. He cut 4 cubes from a potato. Each cube should be a different size. يبقى هنا the changed variable is the surface area. This is what I'm gonna change. Put the potato cubes in methylene blue for 24 hours and then remove the cubes, rinse them in cold water, fill 4 test tubes with water, put a cube in each, leave them for 15 minutes. Some of the blue dye will diffuse out of the cube into the water. Shine a light through the water after 15 minutes. Measure the percentage of light that is absorbed in each test tube. يبقى أنا هنا I did a different experiment. I changed the surface area And what am I measuring? ايه results that are measured? Percentage of light absorbed. Therefore, what is the independent variable? خد بالي برضو ان هنا ما قاليش independent و changed، لا هو قال لي independent بس. لازم ابقى عارفة ال independent variable is the factor changed. What did I change? I changed the surface area. I used different sizes. طب ال dependent variable, what is the results you measured? ال results كانت ايه؟ I measured the percentage of light that is absorbed by the blue dye. طيب, the higher the concentration of the blue dye in water, the greater the percentage of light absorbed. هو ده الايه؟ ال conclusion يعني. State one variable that was kept constant in the investigation described. ايه اللي انا عملته زي بعض؟ مثلا اهو after you put the potato, you remove the potato, rinse them in cold water, fill the test tubes, place one, leave them for 15 minutes. يبقى the time of soaking in the methylene blue كان زي بعض. I soaked them all for 15 minutes. Also the time left for the dye to diffuse, you left them all for 15 minutes برضو ممكن. This is the constant variable. طيب, figure 1.4 is a graph showing the students results. الsurface area اهو ده results بتاعته, surface area and the percentage of light absorbed. What does he want? Estimate the percentage of light absorbed when the surface area was 18 centimeter. You have to show on the graph, so you will draw a line عند 18 centimeter and then go left in order to read what was the percentage of light absorbed. فطلعت لي كام؟ طلعت لي 68%. Okay. A scientist investigated the nutritional content of the fruit juice. State the name of the reagent that would be used to test for starch. This is also food tests. Iodine solution. طب ايه reagent to test for reducing sugar? Benedict solution. Benedict reagent. A sample of fruit was tested for protein. What is the positive result? You can either say purple or lilac. And then الديزاين الاكسبيرمنت بقى. Fruit juice contains vitamin C. Plan an investigation to determine the effect of temperature on vitamin C concentration. Same steps. Number 1, الريبليكا. How many test tubes will I use? I used 5 test tubes. What did you change? You changed the temperature. عايزة أشوف ال effect of temperature، يبقى كل واحدة هحطها في temperature مختلفة. 10, 20, 30, 40, 50 degrees Celsius. يبقى number 2 ال changed variable. Number 3 ال fixed variables. Same type of juice, same volume of juice, same concentration of DCPIP, which is the reagent to test for vitamin C. What results are you going to measure? I will measure the number of drops of DCPIP for a color change. Our volume of DCPIP to see a color change. مش لازم ال number of drops. طيب, انت you will put the juice in different temperatures, but each temperature should be maintained. لازم ال temperature to stay at 10 degrees. Temperature stays at 20 degrees. So how are you going to maintain? Number 5, ال improvement. Maintain the temperature using thermostatically controlled water bath. And then safety precaution, wear goggles, repeat twice, and calculate an average. Okay. Scientists wanted to find out if drinking beetroot juice lowers blood pressure. Two groups of men had their systolic blood pressure measured. Group 1 drank 5 cubic centimeter of juice. Group 2 5 cubic centimeter of apple juice. يبقى ده beetroot وده apple juice. After 6 hours their blood pressure was measured. Okay. Using the data, calculate the percentage change and give your answer to the nearest one decimal place for group 1. هعمل ايه؟ difference between the two numbers over the original number times 100 and then round up to one decimal place فتطلع لي 3.8. State one factor that was kept constant. ايه اللي الاتنين عملوه زي بعض. Both of them drank the same volume of juice 500 cubic centimeter. Okay, هنا بقى photo micrograph of a section through a bronchiole. How am I going to draw it? أولا count الprojections 1 2 3 4 5 6 7 8 9 10 11 12. Okay? And then you have layers. في this dark layer and a light layer and there is another layer هنا. طيب يبقى أنا لما أولا he asked you to do the magnification. Line PQ represents the width of the bronchiole. Measure PQ. Same thing, you will measure it with your ruler in centimeter and then you will multiply by 10 in order to write it in millimeters. Calculate the actual width. Put the triangle. I want the actual width, so I will write image over magnification. Where is the magnification? It is under the photo. يبقى الimage, what you measured, over the magnification, which is 130, and then put the actual here. Make a large drawing of the layers of tissue. This is what I did. أنا رسمت الprojections like the drawing. الprojections, there is a dark layer. This is the dark layer, and then there's a light layer, and there's an outer muscle layer كده. يبقى الfour marks: clear continuous lines, no shading. sorry. ماشي، ما هو لسه هقول في الماركينج سكيم there was a range برضو. يعني انا قلت لكم لما بيكون في حاجة كتير الرينج كان من 10 ل 12. The range of projections was from 10 to 12. 1 mark for the large drawing, no shading. 1 mark for the appropriate layers appropriate layers with number of projections. The last question: a student investigated the effect of exercise on the breathing rate and the heart rate while running. They measured the percentage increase in the breathing rate and the heart rate. The results are shown. These are the results. عايز ايه؟ plot. Use all the data to plot a line graph. You will need to plot the breathing rate and the heart rate as two separate lines. طيب, the first thing, put the labels left side of the table on the x-axis, the results on the y-axis. These are the labels I have. The numbers are in equal interval: 60, 120, 180, 240. So I can put them as they are: 0, 60, 120, 180, 240. The y-axis أنا عايزة numbers until 145 مثلا. فأنا حطيتها ازاي؟ اهو 0 20 40 60 80 100 120 140 ودي هتبقى 150. هي نص بوكس، فال 145 هتكون هنا، ال 142 سوري هتكون هنا. طيب how do I plot the two sets of data؟ ولا أي حاجة. I just plot the two lines separately. بس for this line I put the points as x's, while for this line I put the points as a dot and a circle, and then join point to point using a ruler. وبعدين I will put a key. X ده ال breathing rate, ال dot and the circle is the heart rate. اهو this is how it should look like. Yes, مش ده line graph؟ line graph using a ruler. Okay, point to point, point to point using a ruler, point to point using a ruler. State the conclusion for the data shown. Exercise increases both the breathing rate and the heart rate. Both of them increase with exercise. And this is it for March 23.